The next thing I want to demonstrate to you is how do we go about preparing a slide to use in the lab. Well, there's several different techniques that we can use. One of them is called making a wet mount. So a wet mount is made by taking a clean glass slide and adding a drop of water. So I have a drop of pond water that probably has many little different microscopic organisms in it, and I'm going to place 2 drops on the slide. And I'm then going to place a cover slip over the top of it. So the cover slips are very fragile, they're made out of glass, and you should handle them by the edges to prevent fingerprints showing up on the glass. So make sure they're clean, and when you place it on your glass slide, you want to simply hold it on a 45-degree angle until the water touches the edge of the slide or the cover slip, and then drop it down gently to prevent any type of bubbles from forming. So I just made my first wet mount. I now can take the wet mount and place it on my slide. I can use my stage clips and click it into position. And of course, I will start with my coarse adjustment and try to find the image that I'm looking for. So once you're focusing on the image and you find it under scanning power, then you can adjust to higher magnification as well. Sometimes you are asked to put larger objects onto a slide in order to view them. So here's the other practice to use when you're making a wet mount of something that's a little bit larger. Place a drop of water on the slide, and then take the object that you're going to view and either tear off or cut off a small piece. So let's say I want to look at the leaf structure of this particular plant. I can take a scalpel and cut a very small piece of this plant leaf. So cut off a small piece, and instead of using your hand to try to handle something so small, use a forceps, place it right on top of the drop of water, and then again you go through and take your cover slip and place it over the top of the specimen using the 45-degree angle until it hits the drop of water, and then drop it on there. So you'll notice that there is some room or gaps underneath the cover slip. So if you need to add a little bit more water to your slide because it's not fully submerged, add the drop of water to the side of the cover slip, and it will pull the water underneath the cover slip, and then you can view this underneath the microscope. Now, one of the things that you need to be cautious about if you're trying to view something under high power, when you have a cover slip on, you want to make sure that you view from the side when you're switching to high power. So I can find the object under scanning power. So there's the leaf structure, and I can see lots and lots of leaf cells. I can then adjust to low power, and I can bring it into focus. And then if I wanted to go to high power, I can simply adjust to high magnification, and you'll see that there's not much of a gap between my cover slip and my slide. And now I specifically use the fine adjustment knob to bring in the specific details that I'm trying to identify. So there's a quick overview of how to use a microscope in biology class. You'll be using these tools all year long, so make sure you are acquainted and speak the language of a biologist. Thanks very much.